Good morning, everybody. Happy Mondays. Okay, give a couple of minutes, people roll in, or any questions that anyone has, feel free to ask. All right, let's get cracking, as they say. Let's go back up to here. Okay, so job for today is to finish off DNA replication in eukaryotes. I'm sorry. Which is largely similar to prokaryotes, but with a few added wrinkles. Essentially, the key differences between, let's put the chat up. Right, key differences between procs and eucs, because I'm too lazy to write out prokaryotes and eukaryotes, are one, eucs have chromatin. So histone has to be, or histones have to be removed, synthesized, right? Because you need twice as many as you did to begin with, because you're going to have two lots of chromosomes, two lots of double strands, right? And so you're going to need twice the amount of histones as you did at the beginning. And then they have to be put back on again. Right? Once DNA replication has finished and you've done all of the fixing the RNA primers and gaps and sealing the backbone and stuff like that. There are some small differences in DNA poles. For example, there's no primase. Right, one of the DNA polymerases is actually able to do that. Don't really know how, it's kind of curious. Oh, puppies are having at it again. What else? 3, euchroms are linear. And that equals problems with the ends. I mean, obviously, that also means that you're going to have multiple replication bubbles per chromosome rather than just the one, which you do in prokaryotes. That's obviously another difference as well, but that's kind of self-evident. And so we're also going to talk about the end problem. And that's all about telomeres and telomerase, which is kind of neat as well. So we should be able to get through all of that today without too much trouble. And then if we have time at the end, pardon me, we will talk about, to get started on homologous recombination, which is kind of a neat process, actually, in a lot of different ways. Okay, so just again, as a recap, eukaryotes have chromatin. Chromatin is DNA and protein, right? Prokaryotes do not have that. Part of the reason is that prokaryotes don't have a packaging issue that eukaryotes do, right? Their DNA is kind of present throughout their cell, even though it's kind of concentrated more in one area than another. Whereas eukaryotes have a much larger genome and a smaller space to put it in. So to get around that problem, still be able to use that DNA, eukaryote DNA is packaged using proteins, essentially wrapping it around histones to like spools, like kind of cotton or wool around bobbins, right? So that it takes up less space. Ooh. Oh, I'm sorry. I'm trying to stay awake for the rest of the lecture. Okay, so eukaryote DNA polymerase is a wee bit more complicated than prokaryote ones. The key difference is that in prokaryotes, you have one DNA pol 3, which does all of the elongation, and DNA pol 1, which does removing and replacing primers, right? So DNA pol 3 does both leading and lagging strands. It continues after primase has started, the RNA primer. So in eukaryotes, instead, we have a funkier system where there are 3 polymerases that do most of the work, right? However, instead of one polymerase doing the whole job, essentially it's split up into both beginning and continuing leading strand and continuing lagging strand, right? And so for both strands, DNA polymerase alpha both synthesizes the primer, which is pretty funky. I wasn't entirely lying to you when I said DNA polymerase can't do that. It was just prokaryote DNA polymerase can't. I actually need to read into this more to kind of figure out how it does that. But this is still, essentially, it's a slightly an RNA polymerase too. This is why I got to read into it so I can really fully understand what's going on here. I'll make a note of that. Read up on DNA pol alpha. There you go. Try not to wash my hand before I get to that. And so it basically starts the job, both the leading and the lagging strand. It synthesizes a primer, and it, from memory, it synthesizes, I don't know, a few bases past that, right? In the tens of bases, I believe. It's not a long distance polymerase. And then on the leading strand, DNA polymerase epsilon, with the E, you see there, picks up and runs with it, and that synthesizes the rest of the leading strand. And I think that's delta, does the same for the lagging strand, right? So it's not as simple as with prokaryotes. And that's kind of a general theme in biology overall. By and large, at least the level that we're looking at, both eukaryotes and prokaryotes have the same general mechanisms, right? Because the problems are largely the same that need to be overcome. But, sorry, my wife's having a go at one of my daughters. Must focus. But eukaryotes are generally a little bit more complex, right? They typically have more genes doing the same task, more different proteins and kind of different subtleties about how they do that. Oh, she's not very happy. Uh-oh. Oh, getting the look. I shall run away now, never to be seen again. Yeah, so eukaryotes are just more complex in general, right? They have more more things involved. They have extra kind of funky wrinkles, basically, in how they do the same thing, basically. And part of this also may be the fact that, you know, when you have multiple replication bubbles per chromosome, literally thousands potentially, you're going to need to split up the division of labor, I guess, maybe. Perhaps that's one way of looking at it. And also the issue of removing RNA primers is handled in part by an extra protein called replication protein A. Anybody got any questions about chromatin or DNA polymerases before I move on? Just make sure that that kind of stuff kind of sinks in. As I said when we started this particular section, a lot of this work is really about remembering stuff, you know, what these different things are and what they do, and understanding how they do what they do, what their role is, right? It's a lot more conceptual, right, than what we were working with before, which was much more kind of mathematical logic based. Yeah. Okay, so the same issue applies in eukaryotes as it does in prokaryotes, that they have an RNA primer that needs to be removed, right, and replaced with DNA, and finally having that sugar phosphate backbone resealed, right? So there is no nick in the backbone. And really, we've got the same kind of issue here. You know, we've got our DNA polymerase. This is delta, so that must be for the lagging strand. Yeah, that's right. Oh, I was talking about Okazaki fragments. Of course, we're going to be talking about the lagging strand, even though this does actually occur on the leading strand too. If you remember, if we have multiple replication bubbles per chromosome, you're going to have multiple leading strands per chromosome too. And so, or chromosome as well, obviously not just chromosome 2. But it happens much, much less frequently, right? Because you might have 1,000 leading strands per chromosome, and you might have tens to hundreds of thousands of lagging strands, rather, the Okazaki fragments per chromosome. So anyway, let's use the lagging strand as an example because it kind of makes more sense to talk about that. It's going to happen a lot more there. And we've got our DNA polymerase. It's kind of run up and it's hit the 5' end of the RNA primer for the previous fragment, right? The previous Okazaki fragment. Okay, and so essentially what happens here is that this RNA primer is leveraged out by the replication protein A, right? So before in prokaryotes, if you remember, it's DNA polymerase 1 comes in and actively chews up the RNA primer, right? And replaces it at the same time with DNA. In eukaryotes, it doesn't happen quite the same way. Instead, the RNA primer is levered up and cut off partly into the newly synthesized Okazaki fragment, right? So it's not just the RNA fragment that's removed, it's also a whole bunch of bases past that point, just to make sure. This doesn't actually matter really if you think about it, because all of these bases are going to be replicated. So again, another wrinkle here is that this DNA polymerase has paused, right? It hasn't left. In prokaryotes, the DNA pol 3, once it hits the 5' end of an RNA primer, it falls off, goes and does something else, whatever that might be, synthesize another lagging strand maybe. Okay, so it's up to another DNA polymerase to come in and can, you know, finish the job essentially. In eukaryotes, the DNA polymerase that was synthesizing this strand here, right? So this top one is the daughter strand essentially. It pauses, it doesn't leave. So that once that RNA primer and bit of DNA has been removed, DNA polymerase delta can just continue. It's almost like it's just waiting. It's like, okay, all right. And then it kind of goes on forward, right? And once it hits the very end, then it falls off. I don't know how it can tell that. Maybe it can tell the difference between RNA and DNA nucleotides. I'm not really sure. Again, these are some of the things that I need to kind of read up on. And once it falls off, then the same as in prokaryotes occurs, which is DNA ligase comes in and seals the nick. Right, so this is one of those kind of subtle differences, I'd say, between prokaryotes and eukaryotes in terms of how stuff is done. The same problem is there, right? The need to remove the RNA primers and reseal the backbone. It's just how that is done is a little bit different, right? So that's, you know, kind of a good illustration of how you can get those differences between prokaryotes and eukaryotes. Okay. Oh man, I should have a coffee before teaching. All right. Any questions? Oh, this is also a good chance for me to have a drink of water. Okay, so these RNA primers also lie at a pretty major issue with linear chromosomes. If you think about this, right, we've got DNA polymerase coming in. This is a kind of a good way to illustrate the issue. DNA polymerase coming in, boop-a-doop-a-doo, removes primer, continues on, fills in the gap. Okay, that will make sense. What if that RNA primer is at the very, very end of the strand? So if you kind of put up your hand and just kind of block off where that DNA polymerase is on the screen, and this RNA primer is at the very, 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 very end of the chromosome. Because obviously there has to be one there, right? Because otherwise you won't be able to synthesize the DNA kind of at the very, very end of the chromosome. So this RNA primer has to be removed because you can't have RNA in your genome. How do you fill in that gap? That's a question to the class. How can you fill in that gap if there's nowhere there's no 3' OH for DNA polymerase to use? Well, the answer is you can't. You always have to have a 3' OH to continue, or no, begin DNA synthesis. Problem, there is no 3' OH at the very end of the chromosome. And so that means that that gap does not get filled. You might think, okay, well, that's not that big of a deal, right? But the next time that cell divides, the end of the chromosome will begin here. And so the next time there's going to be an RNA primer here, kind of underneath this box, and that gap isn't going to be filled. So every round of cell division, your chromosomes get shorter, right? And this is kind of illustrated quite neatly here. If this was one whole chromosome, basically and this is just kind of blown up down here for illustration's sake, every time you copy this chromosome, it will get shorter by the length of that primer. And that happens over and over again, over and over and over again. Okay, so there's a couple of things going on here. One, we need a buffer at the end of each chromosome, or both ends actually, right? So that that shortening doesn't actually chew into functional genes, right? Because you can imagine if this primer where that was removed is part of the, I don't know, the regulatory sequence of a gene, right? And it's removed, now that gene will not get turned on. Or if it goes far enough, even worse, it starts like removing the start codon of that gene. That gene will never get synthesized, right? It'll never get turned into a protein, right? Which is really, really serious. So this is called essentially the end replication problem. So kind of like a, I don't know, sticker plaster kind of or band-aid type solution is to have um, a buffer at the end, at each end of the chromosome. And these are called the telomeres, or telomeres, telomeres, mirrors, like mirrors in American, but mirrors. Anyway, these essentially are highly repetitive DNA. And they don't code for anything. So it doesn't actually matter that these telomeres get shorter. Actually, if you look at my chromosomes, for example, if you kind of took them out nicely and actually measured the length of them by sequencing or some other method, I'm not sure how exactly, you would find that they're quite a bit shorter than those of my daughters. Even though my daughters each got half of their chromosomes from me, those chromosomes are longer than mine are because mine have gone through more rounds of cell division, particularly in cells that are more proliferative, right, that go through more rounds of cell division, such as hair, skin, gut, lung, any kind of epithelia, basically, any kind of surface type cells. And so as you get older, your chromosomes get shorter, right? But there's a limit. And that's basically when, I mean, that buffer doesn't last forever, right? At some point, you're going to get close to the end of it. And so now you need to say, okay, this cell, you can't divide anymore, right? You've used up all of your buffer, and that's it. You're done, right? No more. And that's called the Hayflick limit. And that equals around 60 cell divisions, which sounds like a lot, right? But when you consider that your gut cells are being replaced continuously, your hair is always growing, your skin is constantly being replaced, it doesn't actually sound like an awful lot. And so now, and this is after essentially once a cell hits the Hayflick limit, it stops dividing and enters what's called senescence, if I can spell that right. Right, which is basically enforced aging, which is rather sad. And so those cells kind of stop functioning quite as well. They don't die, they just hang around and kind of don't do very much, essentially. And that's actually part of organismal aging, right? So if you look at young adults or teenagers, they have very, very little cellular senescence. If you look at someone like me or my granddad, or my dad rather, not my granddad, is hasn't been around for quite a while. But my dad, he'll have considerably more than I do, right? And that's part of why, our hair goes gray, why, because pigment producing cells don't work very well anymore. Why lots of things don't work quite as well as they used to, which is a sad topic to talk about. We aren't going to talk about that anymore. We're going to live forever, right? As they say in Starship Troopers. Classic movie quote there. Anyway, it's a brilliant film if anyone's ever watched it. Completely ridiculous, but really funny. So you might be thinking, whoa, hang on there. There are clearly cells which escape that limit, right? Two types of cells, one that's good for you, one that isn't. All of your from your own cells, not like outside pathogens or whatever. What cells escape the Hayflick limit? Can anybody think? Two types of them. Cells which can essentially divide. No, interesting answer though. These are, sorry, I should have been a little bit clearer, somatic cells, right? So these are cells which, I mean, actually, that is a really good answer. In females, no. In males, however, you are continuously producing sperm, and so even to kind of like your dying day as a like 80 or 90-year-old. And so the cells which generate the cells which produce sperm must also be immortal, essentially, right? Able to divide continuously. So yes, actually, that thinking through it, that was correct. Crap. Okay, 3 lots of cells then. What are the other 2? Same idea though. Think about cells in your body which essentially never die and keep reproducing, other than gametes. I already talked about some of the tissues and where you find them. It's an acronym that's often used to describe the sciences and engineering and mathematics type fields. There you go. Stem cells. And so these can divide endlessly and without limit. And they're essentially what they do is they provide new cells for, I don't know how one would describe it, replacing tissues. So when your skin is damaged, your fingertips are kind of cut off, your liver is damaged and stuff like that. You need to generate cells which can divide again, right? And you can either turn the clock back in terms of developmental time, called dedifferentiation, or you can use stem cells. And so typically stem cells are what are able to generate new skin cells, new hair cells, new intestinal cells, right? Your intestine is actually a site of very high cellular turnover, as it literally gets abraded. And so these stem cells, what they do is they essentially divide. You get two cells. One of those goes back into the stem cell pool, typically, and the other one can then change and differentiate into whatever cell it needs to be to kind of keep that process going. Right, so stem cells are one. Can anybody think of another type of cell which divides continuously and is generally very bad for you? One of your own cells or your own cells from your own body. Something we can, yeah, cancer, exactly. Fabulous answer, generally. So cancer cells can escape that limit as well. I mean, you can actually, there have been in like World Guinness Book of Records cases of where people have tumors that weigh as much as they do, right? And that can only occur if those cells are able to just continually divide without any check, right? And so you're probably thinking, well, why doesn't this end replication problem apply to stem cells and cancer cells? And really the cells that generate new gametes are also stem cells as well. They're just a different type of stem cell, right? And so the answer is, and it's called escaping the Hayflick limit, which I think is kind of cool. And they do so by repairing their telomeres. And they use an enzyme called telomerase. I'm not going to go into the mechanism of how that works because it's kind of funky, but it uses those repeat sequences to essentially extend the chromosome a little bit, use an RNA, and then use that RNA, which kind of loops back to fill in the gap that was left by the removal of that primer. And so in these cells which are expressing telomerase, right, this end replication problem does not occur, right, because these gaps are filled. I think they can even rebuild the telomeres as well. I'm not 100% sure about that, but I think they can too, right? And so control of expression of telomerase is very tightly controlled because you really do not want this to be working in cells which aren't stem cells. This actually, the activation of telomerase is a key characteristic of cancer. Oop, okay, didn't see that. There you go, cancer. So cancer is obviously caused by a lot of different things, right? Lots of things have to go kind of wrong at the same time for cancers to form, right? But one of the things which has to happen, whatever the other causes are, is the activation of telomerase. Because otherwise, you would have a cancerous cell which can divide, but it can only divide a fixed number of times, right? Until all of its daughters hit the Hayflick limit and cell division stops, right? So it'd be self-limiting essentially, right? You would never get beyond like, little teeny tiny tumors which don't really necessarily cause any problems, right? So anyway, I thought that was kind of like an interesting aside to add in. So this is, but it's also a very difficult thing to target as well. So even though it has to happen for cancer cells to escape that Hayflick limit, you can't target it as like a drug target in cancer treatment because all of your stem cells also need that telomerase to work, right? And so that's kind of one of the very, very difficult aspects of cancer biology and treating cancers, is that they do in many ways actually appear very much the same as you, because they are you, right? They're bits of you which shouldn't be there, but they're still made from you, right? And so trying to kind of differentiate between, cancer cells and other types of dividing cells is very, very difficult, right? And that's why often when you give patients chemotherapy, right, which targets fast replicating cells, they lose their hair, right, and have gastrointestinal problems and uh, immune system problems as well, because all of those things, all of those tissue types are also targeted by the same chemicals that target cancer, right? So that's kind of a good illustration of how difficult that problem is. Okay, so before we move on to homologous replication, does anybody have any questions about any of this? Okay, awesome. So I want to talk about homologous recombination for a couple of reasons. One is that it's pretty neat. Well, actually, more than two reasons. One is it's something that has to happen in cell division. Specifically meiosis, right? And this is how we generate new combinations of alleles in our gametes, right? Which will go into the next generation. And it's also a way in which you can repair DNA, like really serious DNA damage, right? And so this is actually a kind of like gold standard Cadillac version of fixing double-stranded breaks. Just as a double-stranded break, or DSB. Which is where both, where the backbones of both strands in a double helix are broken. And this is pretty catastrophic, right? Because for the same reason why you don't want to have RNA primers or unligated backbone, right, after DNA replication, because next time you open that DNA up, it will fall to pieces. It's even more so an issue for when you break both strands. And the problem with breaking both strands is that how do you know what end goes with which, right? Maybe if you just broke it in one place, it isn't too bad. If you break it in two places, how can you tell what end goes where? It's like cutting a piece of string in multiple places and then going, okay, which end goes with which, right? I mean, there's no way of knowing. And that's the kind of damage that you get from what's called ionizing radiation. So gamma radiation, which is produced by typically radioactive decay, also produced by the sun and stuff like that too, causes double-stranded breaks. And that's typically the reason why you get radiation sickness, right? Is because your cells, particularly highly dividing cells, again, like your immune system and your gut and your hair and things like that, are very, very sensitive to that, right? And so if you get double-stranded breaks in those kind of tissues, you'll see exactly the kind of symptoms you get from exposure to radiation. And so anyway, so this is both a way of fixing those problems, but also is a central part of meiosis as well. And this can only take place when you have essentially 2 copies of each chromosome present. So this can actually take place during mitosis. In terms of DNA repair, it always takes place in prophase 1 of meiosis, right? So it's kind of like every time we have prophase 1 of meiosis, we have homologous recombination, always. In mitosis, this only occurs if you encounter a double-stranded break, right? Because you have to have your 2 chromatids, right? Your replicated chromosome and your homologous chromosome with its 2 chromatids to be able to swap DNA. And this is called homologous recombination because it's going to be between homologous chromosomes, right? That's the only way that you can really accurately do this, because if you were to do it with any other chromosome, well, you know, you just get a complete total mess, basically, right? So this is actually something which is fairly poorly understood, right? It's actually kind of, we started back here, right? I'm not going to go into this in great detail. We'll do that next class because we don't really have the time to do it justice, right? But when scientists were able to get the DNA out of meiotic cells in prophase 1 of meiosis 1, they saw these things called, they're now called holiday junctions, right, after the person that discovered them. And they're like an X shape, right? And so they're joined by these shared bits of DNA, right, essentially, which kind of look like this, right, when you look at it in the cartoon format, right? And these X's, these are very kind of classic X shapes. These are called holiday junctions, right? And so this was how it was originally discovered. And then we kind of spent a lot of time trying to figure out how it works, right? And it's not very well understood, right? It's not very well understood how exactly you can get such a precise alignment of the 2 homologous chromosomes that it's going to be a base by base perfect swap, right? Because if you think about this, right, and this is kind of a central part of this phenomenon, when you swap DNA, you do not want to make a mistake, right? Because it would be very much like if you, I don't know, kind of crossed over 2 sentences, right? And you didn't have those words precisely aligned, you'd end up with a word or a sentence that's shorter than it should be, like so, and another one which is longer than it should be. It'd have extra information. Both of those can be terrible, right? Because you'd either be missing bits of DNA which may encode a protein or be required for turning a gene on, or you might have extra bits which would encode either extra protein or which might not actually work, or an extra bit of DNA which might turn on a gene when it shouldn't be turned on, right? So this has to be perfectly lined up, right? And we think we know there are proteins which do that, which is, shoot, can't remember off the top of my head right now. It's not, it's the synaptonemal complex is what it's called. There you go. I'm just not sure how it does that, right? I'm not sure if it is actually known how it does that. So those 2 have to be perfectly aligned, right? You have to then break both strands, right? Or really, you have to break one strand in one DNA molecule, both backbones, so double-stranded break. Pardon me. And then you have to just kind of like stick it all together somehow, right? So that essentially what you end up with is two molecules joined, right, together, that kind of crossover there, without either losing or gaining any information. And then that has to be cut very precisely, right? So that you end up being able to, kind of separate those later on, right? They're not going to stay stuck together. Obviously, as you remember, in up until metaphase 1, they're stuck together by the cohesin, right, at the ends, like here and here, right, is how those chromosomes are held together. But the actual resolving of the crossover, or the recombination, has already occurred before prophase finishes. So I'm going to just kind of leave it there because this is a bit too big of a chunk to bite off in the last few minutes. Does anyone have any questions about anything that we've covered so far? Okay, one thing that I would strongly recommend if you haven't done so already is make a list of stuff that you need to go through before the exam on Friday, right? Which you should already have, right? Because this is a continuous process, right? This whole studying and revising deal. And bring that with you on Wednesday to class, to this Zoom session, because this is probably going to take, I don't know, 15 minutes, maybe, something like that. And then we'll have the rest of the class for any of those questions, right? Now, that might, if none of you have any questions, that might end up with being a very short class. But I'm pretty positive most of you will have stuff. Not so much a review, because I don't think there's a really good question generally, but I don't think there's really any way that I can go over what would be 3 and a bit weeks of material in 30 minutes. More anything that you don't fully get, or that you want clarifying, or you want to make sure that you understand it, which, we wouldn't tell you telling me what you know, bring that stuff with you, right? And so this would really be a kind of like a study session, right, rather than a review session. So really a way of just polishing you up, essentially, before Friday. Does that make sense? Yeah. So you should all have like a running list of stuff that doesn't quite make sense, that you need to study more, you need to kind of gin up on. Just as a matter of course, right, for any of your courses as you go along, right? Because those are the things you need to study before the exam, more than what you already know. Bring those lists with you, and then we'll go through and we'll make sure everybody gets a chance to ask questions. All right. I think that's probably it for now. I'm gonna go make myself a coffee so I can stay awake. And if you have any other questions in the meantime, feel free to email me them as well, and I'll help out. Cool. That's pretty much it for today. So see you on Wednesday. We'll finish up the last little bit before the exam, and then the exam will be on Friday. So don't forget. Cool. See you on Wednesday, everybody.